Hi everyone, so this video lecture was prepared from the review book of Medical Microbiology and Immunology by Perrin Levinson. So in this video lecture, we will discuss the laboratory diagnosis that include the bacteriologic approach and serologic approach. First of all, we must know that the laboratory diagnosis of infectious disease involves two main approaches. One is the bacteriologic approach, in which the organism is identified by staining and culturing the organism, and the other is the immunologic approach, also known as serologic approach, in which the organism is identified by detection of antibodies against the organism in the patient's serum. So, what are the cultures used in the bacteriologic method? So, first culture is blood culture. Blood cultures are performed most often when sepsis, endocarditis, meningitis, osteomyelitis, pneumonia is suspected. The organism most frequently isolated from blood cultures are of the 2 gram-positive cocci, that is Staphylococcus aureus and Streptococcus pneumoniae. And there are 3 gram-negative rods that are most frequently isolated, that is Escherichia coli, that we call E. coli, and Klebsiella pneumoniae and Pseudomonas aeruginosa. It is important to obtain at least 3 10 ml bottle sample in a 24-hour period. And what is the most important precaution must be taken by a phlebotomist to collect the blood from the patient is the site of venipuncture must be cleansed with 2% iodine to prevent contamination by member of the flora of the skin, that is Staphylococcus epidermidis. The blood obtained is added to 100 ml of a rich growth medium such as brain heart infusion broth, also called as VHI broth, whether 1 or 2 bottles are included, varies among hospitals, and blood cultures are checked for turbidity or for carbon dioxide production daily for 7 days. It's very essential to check the turbidity daily for 7 days or more longer days. If growth occurs, gram stain, subculture, and antibiotic sensitivity tests are performed. If no growth is observed after 1 or 2 days, blind subculturing onto other media. Any revealed organism culture should be held for 14 days when infectious, endocarditis, fungemia, and other infection by slow-growing bacteria, example Brucella, is suspected. So this is the blood culture. Other is throat culture. Throat cultures are usually primarily to detect the presence of Group A beta hemolytic streptococci, that is Streptococcus pyogenes. And important and treatable cause of pharyngitis. Streptococcus pyogene, an important and treatable cause of pharyngitis. They are also used diphtheria and gonococcal pharyngitis or thrush, that is caused by Candida species, then it is suspected. When the species is being obtained, the swab should not touch only the posterior pharynx, but both tonsils or tonsillar foci are touched as well. The material on the swab is inoculated onto blood agar, and if colonies of beta hemolytic streptococci are found after 24 hours of incubation at 35 degrees centigrade, a Bessie-Tristan test is usually used to determine whether the organism is likely to be grown group A streptococci, and if both is inhibited around this, it is a group A streptococci. If not, it is a non-group A beta hemolytic streptococci. Notice that gram staining is not done on throat swab. Why? Because the gram stain is typically not done on a throat swab because it is impossible to distinguish between the appearance of the normal flora streptococci and Streptococcus pyogenes, that is the treatable cause of pharyngitis. Next is stool culture. Stool cultures are performed primarily for cases of enterocolitis. The most common bacterial pathogen cause diarrhea in the United States are Shigella, Salmonella, and Campylobacter. These three are the most causing diarrhea in the US. And gram stain is not done because it is very difficult to identify. McConkey agar and EMB agar is used. Gram stain here is not done because it may reveal large numbers of certain organisms such as Staphylococcus, Clostridia, and Campylobacter. Gram stain of stool is not usually done because the large numbers of bacteria in the normal flora of the colon make the interpretation very difficult. And for culture of Salmonella and Shigella, a selective differential medium, that is McConkey agar or eosin methylene blue, that is EMB agar, is used. These media are selective because they allow gram-negative rods to grow but inhibit many gram-positive organisms. Their differential properties are based on the fact that Salmonella and Shigella do not ferment lactose, whereas many other enteric gram-negative rods do ferment lactose. If non-lactose fermenter colonies are found, a triple sugar iron, that is TSI agar slant, is used to distinguish Salmonella from Shigella. Some species of Proteus resemble Salmonella on TSI agar but can be distinguished because they produce the enzyme urease, whereas Salmonella does not. So that's why gram staining is not done and McConkey agar and EMB agar is used. Next is urine cultures. Urine cultures are performed primarily when pyelonephritis or cystitis suspected. By the far most frequent cause of urinary tract infection, UTI infection, is E. coli. Other common agents are Enterobacter, Proteus, and Enterococcus faecalis. Urine in the bladder of a healthy person is sterile, but it acquires organism of the normal flora as it passes through the distal portion of the urethra. To avoid these organisms, a midstream specimen. Noted that a midstream specimen voided after washing the external orifice is used for urine cultures. In special situations, suprapubic aspiration or catheterization may be required to obtain a specimen because urine is a good culture medium and it is essential that the culture be done with 1 hour after collection or stored in a refrigerator at 4 degrees centigrade or more than for 18 hours. And it is commonly accepted that a bacterial count of at least 1 leg per ml. 1 leg per ml must be found to conclude that the significant bacteriuria is present in asymptomatic persons. There is evidence that as few as 100 per ml are significant in symptomatic patients. For this determination to be made, quantitative or semi-quantitative cultures must be performed. So let's summarize the whole discussion. The laboratory diagnosis of infectious disease include bacteriologic, immunologic, serologic, and molecular, that is nucleic acid-based tests. Bacteriologic tests typically begin with staining the patient specimen, observing the organism in the microscope. This is followed by culturing the organism, typically on blood agar, then performing various tests, obtaining by pure culture. Blood cultures are usually useful in cases of sepsis and other diseases in which the organism is often found in the bloodstream, such as endocarditis, meningitis, pneumonia, and osteomyelitis. Throat cultures are most useful to diagnose pharyngitis caused by Streptococcus pyogenes that we discussed earlier, strep throat, but they are used to diagnose diphtheria, gonococcal pharyngitis, and thrush that is caused by the yeast Candida albicans. Stool cultures are used primarily when complaint is bloody diarrhea, that is dysentery, enterocolitis, rather than watery diarrhea, which is often caused by either enterotoxins or viruses. Routine cultures are used to determine the cause of either pyelonephritis and cystitis. General genital tract cultures are most often used to diagnose gonorrhea and chancroid. This is all about the laboratory diagnosis approach of bacteriologic approach and cultures that are used in a laboratory methods. Let's cover the general approach to the diagnosis of bacterial infection. Thanks for watching. I hope you liked this video. Hit the like button, subscribe my channel. Thanks for watching.